Okay, great. So yes, again, thanks everyone for being here. I'm very delighted to be kind of kicking off this seminar series, and I hope you will join us for the remaining seminars in the coming weeks. So this talk is about knowledge-based planning, and I'll talk about what that means a little bit, but it's going to be a mix of around prediction optimization with applications to medicine. And we'll talk a little bit about open access as well. So I'll start kind of backwards. I'll start at the end and give acknowledgements first. And there's a long list of people to acknowledge in the work that I'm presenting today. I'll be presenting multiple papers. And but if I had to really single out one person, it would be Aaron, the name at the top there. So Aaron was a former PhD student of mine. And most of the work that I'm talking about here is really, all of the work really that I'm talking about has really been led by him. He did an amazing job over many years, and then working with other students and then collaborators in the hospitals and many others that you'll see in a second. Okay, so here's the overview of the talk. I'm going to talk about essentially three papers and sort of three mini talks in this one seminar. So the first one around knowledge-based planning, so I'll give you some background, and then I'll talk about a couple of challenges that we organized and kind of bringing together people internationally to solve some of these problems together. Okay. So the focus on the kind of medical motivation for this work is cancer. Okay, so if you have cancer, there are a few ways to treat cancer, a few main ways. There are many ways, but kind of the most common ways that people know about, that they've heard about are, surgery, you cut out the tumor. Chemotherapy is where you inject fast-acting drugs into, you inject drugs into the body to kill sort of fast-dividing cells, right? And then the third main way is radiation. And so here's a picture of a linear accelerator. This white object is the linear accelerator. It shoots radiation out of the head here, and the patient lies on this table. And this whole white object, you can see it's on an angle, so you can rotate it 360 degrees essentially around the patient, and you can therefore shoot radiation from multiple angles. And the purpose is so that where these beams intersect, you hit the center of the tumor, hopefully, and you deliver less dose to the healthy tissue all around the tumor. Okay. Treatments are typically what are called fractionated. Fractionated just simply means you deliver the treatment over many days, over many weeks. So you spread out the radiation delivered to the patient, give them a chance to recover. And this takes advantage of something called the therapeutic advantage, which healthy cells have that cancerous cells don't. Healthy cells have the ability to repair themselves a little bit. So that's why we want to spread out the radiation, but that still doesn't hurt the treatment because the cancer cells don't have the opportunity to repair. So the focus of our work is really around how do we design or optimize these types of treatments, okay? So to understand that, we need to understand a little bit about what the planning process is. So at a high level, the process to create a treatment of radiation would be that we take CT images of the patient. So here's an example image of a cross-section of a patient where the tumor is basically this yellow object in the middle. And then you see there are a few other healthy structures nearby. So this is the bladder, this is the rectum, this is the left and right femur bones, so the thigh bones. And then, and this would be a tumor in the prostate of the patient. And so we take an image, a doctor will look at this image and will contour the structures. We'll say, okay, here are where the healthy structures are. And, these days, thinking about machine learning and the focus of the seminar, right, there's more and more research that's being done in methods that you can use deep learning methods to automate the contouring and identification of these structures, right? But that's not the focus of our work. Once we have these structures that are contoured, we use this as data in an optimization model to decide where to deliver the radiation. So you see in this picture, there's 7 beams, 1, 2, 3, 4, 5, 6, and 7. So coming from 7 different angles, for example. So optimization would look at things like, the angles that we're shooting from, what does the intensity profile look like from every angle? There's other treatments that deliver radiation continuously as you rotate in a continuous fashion around the patient. So all of these are different types of optimization models, essentially. Once you create the first treatment, you will calculate some metrics around the goodness of this treatment, and then the planner will show it to the oncologist and get their feedback and iterate. The plan might be, oh, there's too much radiation here and not enough there, so there will be some back and forth until we arrive at a final treatment that everyone is happy with, and then we go deliver it to the patient over multiple weeks. Okay. To give you an idea of what the optimization variable looks like, so here, imagine this is a tumor, and this OAR, this green structure, is an organ at risk, so a healthy organ that we'd like to avoid, and we're delivering radiation from 3 angles. So this distribution here represents the profile of the intensity of the radiation. So basically says at this part of the beam, we're going to deliver more radiation, and at this part of the beam, we'll deliver less. Same thing here, less here, more here, more on 2 sides and less in the middle. And the idea is that this profile can be optimized so that when you sum up the radiation delivered from this beam and this beam and this beam, you get a shape of radiation that conforms to the shape of the tumor that hopefully has high uniform dose, let's say in the red region, and very little dose outside. Right? So you can imagine that this is a very complex problem, and we really need something like optimization to help us figure out what these profiles ought to be. Okay, so the reason I show this picture is to help you understand the decision variable in this problem is the intensity profile, basically, for each of these beams. Okay. So everything that I've described up until now is kind of background was relevant, 20 to 30 years ago. So we would take an image, we would, draw on the image the structures that we want to deliver radiation to, the structures that we want to avoid, and then the optimizing of the beam based on the image. Okay, and the key is that this process would iterate potentially multiple times. And for very complex tumor sites with lots of organs at risk, you might iterate many, many times until you get at a solution. And then where this field moved to over the last 10 to or so years is around what we call knowledge-based planning, is to try and avoid this iterative process of planning and replanning, but to try and do it in one shot, mostly in one shot, okay? And so here, the linear approach that has been proposed looks something like this. So we still need to contour the image. We still need to decide, to identify where all the structures are. But then instead of optimizing and then re-optimizing over and over, we make a prediction. We use the power of machine learning to predict, using all of the historical data that we have gathered up until now, we make a prediction of what a good dose distribution should look like, or what a good plan should look like, because we already have, labeled data, right? We have the X's, which are the images, and the Y's, which are, think of it as like the dose distribution of the plan for hundreds, if not thousands, of historical plans that were approved clinically and delivered to the patient. Okay, so we can train a model to map these images to the dose. However, this dose is simply a 3D10 sort of numbers, right? It doesn't tell us anything about how the machine should deliver the radiation to achieve this dose. It doesn't give us the parameters of the machine to deliver it. So we would run this prediction back through an optimization process. And so the optimization model now is really to convert the prediction into a deliverable treatment. And so it gives us instructions on how to deliver essentially this prediction. We try and come up with a plan that is, in some sense, as close as possible to this prediction, and then we proceed with the process of delivering the treatment. Okay. So I hope that's clear. So we're trying to reduce these iterations and go straight from our historical knowledge to a prediction to then an optimization. Okay, so this process we'll call knowledge-based planning. Okay, so just for some background, some literature. So this topic has been explored for quite a long time now. You see, literature going back well over a decade. And so early work would use more classical ML methods to predict a dose volume histogram point. So think about a distribution of dose like this, okay, and think about a histogram based on this distribution, and you could predict something like, the 90th percentile or the 80th percentile or something. So you could try and predict like some sort of percentile of the distribution. And then when people started to really realize the power of using deep learning to CNNs, for example, to make good predictions from image-based data, then people started to directly try and predict this 3D spatial dose distribution directly from the image without trying to compress it and change it to a linear model or anything like that. So again, some of the earlier work was looking at predicting the 2D slices independently. So thinking about a 3D object, separating it into one slice at a time, and then predicting a dose distribution one slice at a time, but not thinking about the correlations across slices. And then even, and then obviously more recently, trying to predict the full 3D dose distribution all at once and taking into account those vertical correlations. So our approach, I think, is one of, I can't remember if it's one of these papers, it might be this one, where we started to look at, this was basically when GANs were becoming very hot. And so we looked at using a generative adversarial network to predict dose distributions. So we would take a CT image, we would feed it into essentially a neural network to make a prediction. It would generate a dose that would be evaluated by another neural network, a discriminator. And this artificial dose would be compared against the true clinical dose, because we have the ground truth label, and it would try and discriminate between which one was the real and which was the artificial, and then send back information to this first network to then revise its inference to generate a new dose distribution and so on. And the interesting analogy here is it actually kind of replicates the traditional approach to creating a plan, right? We'd have someone design a plan, we would show it to the oncologist, they would give feedback, and then they would revise, right? So the idea is this pair of neural networks is basically doing this process. And then finally, when we get to a point where we find the performance sufficient, then we have an ability to create or produce realistic looking dose distributions. Okay, so we train the networks to do this. And then once we have fully trained the network, we just simply use the first one, the generator, to now take a new image that we haven't seen before, run it through, and then generate a dose distribution. And then now we use this generated or predicted dose distribution to feed into the optimization process to generate the treatment plan. Okay. Just checking the Q&A. I don't think there are any new questions. So yeah, feel free to ask questions as we go. Suman?
No, they're not. Sorry.
Oh,
I said
okay.
no new questions.
Okay, thank you. Okay. So we developed, we started with a 2D GAN approach, which was the idea that we would look at each slice of the image independently, and then use the CT image and try and predict a dose distribution. And then we eventually developed a 3D model where we were taking the full 3D image, so basically a collection of slices, knowing that the first slice is on top of the second slice and so on. Excuse me. And try and predict the full 3D dose distribution all at once. Sorry,
so
what was that?
There's a request to show the previous slide again, if possible.
Mm-hmm. This slide or a different one?
I think this slide. Oh no, the previous one.
One more. Okay. This one?
I guess so. Ali, who is asking, is that the correct one?
Okay. So yeah, if you have a question, I'm happy to entertain it. But in the interest of time, I'll keep going. And if you, I mean, there will be a recording afterwards, so you can definitely take a look at it further. Okay, and so just to give you a sense of the 2D versus the 3D, right? When we make these 2D predictions, so each one of these slices vertically is independent, and what you see is there do seem to be some unrealistically sharp gradients there, right? So the dose goes from very low, which is blue, to a bit hotter, and then boom, all of a sudden it jumps to this orange level, which is quite hot. So that's a bit unrealistic, and that's we see the same thing here, we see the same thing here, and this is certainly a result of ignoring the vertical relationship. When you look at the 3D approach, that doesn't have that, right? And so how do we then evaluate these models? So we take the CT image, and then we are going to compare our 3D model to a 2D model. So these are the two GAN models. And we will also compare it to some existing baseline models that exist that, for example, wouldn't be deep learning models and so on, or other deep learning models that are not GANs. Then we'll make predictions from them. These predictions will go into the same optimization method. So we hold this part constant, and then we get plans out of this optimization model. And the idea was basically to compare the quality of the plans based on the different types of prediction models. Okay. So now let me talk about this plan optimization box. What is in this box around optimization? So the optimization box actually itself is a two-step process. So the idea is that in the first step, we would take this predicted dose distribution. We would learn parameters of an optimization model that are most likely to recover this dose distribution. So think about some optimization model where there are parameters that need to be determined by a human, for example. The idea is to basically ask the question, what parameter values will generate for us a dose distribution that is as close as possible to this, essentially, when we do the optimization. So we learn those weights in this process, which we call inverse optimization. And then a bit confusingly, we use those weights in what really I should call a forward optimization. In the medical community, they call this inverse planning. So I'm using their terminology, but it's really taking these weights and then using a forward optimization approach to get the optimal solution. So this would be fluence here just represents the profile of the intensity of the radiation from the different beams. Okay, and then this solution, sorry, I can't get my mouse. This solution is what we would use to generate the dose distribution and so on. Okay, so just again to recap, we take an image, we make a prediction, we get to this predicted dose, we run it through this process eventually to get a solution that returns a dose distribution that hopefully looks as good as the predicted input dose. Okay.
There are a couple of questions. Do you want to answer now?
Sure. Max, I don't know if everyone can see them, but Max has a question. Each person has their own breathing pattern or personal data. Is it possible to incorporate those data in the prediction? Certainly, so breathing is a whole other issue. Maybe I'll put that aside for a second, but the larger question that you're asking is certainly there is some personalization that is possible. I mean, in a sense, like the image itself is already very personalized, right? So it's your personal, the anatomical geometry, where your organs are, the size and shape and so on. There could be other contextual factors. We haven't used any of those in this prediction approach, but it's certainly possible to add that to the prediction. Another question around which model is more accurate. We'll wait a couple of slides and I'll show you that. Okay. Okay, so in testing these models, we got just over 200 real cancer patient cases that were treated at our partner hospital in Toronto. So these are basically head and neck cases. So this region of, so the pharynx is your throat, as you can divide this region into several subregions. And so we were looking at cancers that were really at this part, kind of at the back of your mouth, back of your throat area, the oral pharynx. Okay. So as I mentioned, we had 217 plans. We segmented them into training and testing sets. So we left 130 of them for training and 87 of them for testing. we would train the models. We would then apply the trained neural network model to the out of sample 87, get a prediction, run it through the optimization, and get a plan. Okay, so that's the whole pipeline. When we get the plan, we now compare the performance of our optimized plan to the actual plan that was delivered, right? Because all of these 217 received a real clinical plan from the hospital, and we can look at, for example, how similar they are, how many, is ours better, is ours worse, and so on. So there's lots of different metrics that we've evaluated, and I can happy to point you to the paper to look more deeply. I'll just simply report on one metric to give you a sense. So here I'm going to report on the proportion of plans that satisfy the same criteria as the corresponding clinical plans. So what exactly does that mean? So in a clinical plan, there are many criteria that need to be satisfied. So for example, we would want the brainstem to have a maximum dose less than something. We would want the spinal cord to have a maximum dose less than something. And so you can try and read all of these criteria and get a sense of a bunch of distributional constraints that we want our dose distribution to satisfy. And so for patient one may satisfy this constraint, this constraint, this constraint, and these two, for example. So maybe 6 out of the 9, maybe for one patient, only 6 of the 9 were satisfied. But nevertheless, that was the plan that was delivered. And so the clinician must have made a decision that satisfying those 6 was okay, and not satisfying the other 3 would have been okay because it was too hard to satisfy them all. And so basically what we're saying here is, if we satisfy the same 6 as what the clinician plan satisfied, we would call that 100%. If we satisfied 5 out of the 6, we'd call that 83%, for example. Okay, so we want to compare basically across all of the patients and all of the criteria, what fraction of the ones that they satisfied did we also satisfy. And so here are the results. So here I'm just showing results from 3. So we had a baseline UNet model that was not a GAN. We looked at a 2D GAN, we looked at a 3D GAN. And so I bolded the column that has the highest percentage. So these are all percentages. And so basically what this says is, when we're looking at organs at risk, the baseline model satisfies around 80% of the criteria that the clinical plans do. 2D GAN is a little lower, 78, and this one's 79. So so both of these are 79. When you get to the targets, so the baseline has about 92%, the 2D GAN is about 97.7, and the 3D GAN is 98.8. And so overall, the performance of this 3D GAN, as perhaps we might expect, does perform a little bit better than the 2D GAN, because we are, accounting for those vertical correlations. But when we get about 80%, and this is with, I would say, a fairly straightforward approach. There wasn't a lot of additional tuning or parameterization. It's certainly, like you might argue we could try and do better than that. And certainly I think people have done better than this, but at the time, this was what was, I believe, the best performance at the time. So just the summary for this first part, that, when we were working with GANs, we thought they were very well suited for dose prediction. Certainly better than some of the existing methods out there. I think since that time, the field has moved forward, and people were finding GANs that just be harder to train and a bit more sensitive. And people have now been able to meet these previous results and actually beat it with just a straight up, well-tuned, well-designed, neural network, like a UNet architecture. And I'll kind of mention that a little bit in the second part of this talk, where we talk about a competition that basically all of the models that perform the best were basically like a simple UNet model. One of the important takeaways that we got from doing this study was that all of these results were based on the private data, essentially, that we had access to through our hospital partner. And so we were doing this in Toronto, and other people were doing this in San Francisco, and other people were doing this in New York, and so everyone's working on their own little data set, trying to develop the similar model to get similar or better performance, right? And so the challenge is external benchmarking is next to impossible when we're all working with private data. So we decided to organize a competition, which we called the Open Knowledge-Based Planning Competition, to try and encourage more collaboration internationally and sharing of results and having a benchmark to compare against consistently. So
we
I'll
actually
maybe just
have a
pause there. I know there's a
question. Yeah, we have a question that might be actually relevant.
Okay.
I don't think everyone can read, so if you could read it out.
Yeah, I will. Let me just read it,
Sure.
and then I'll read it out to everyone. Yeah, it's a great question. Thank you, Kamawu. So the question, the comment is, I'm not an expert in dose prediction, but I guess the data set might be limited and severely imbalanced. I wonder if the proposed optimization and planning would work well on minority data sets, for example, specific ages or genders, as well as the majority data set. Yeah, it's a very good question. So for example, for this data set, we didn't take note of, I don't even know if we had, access to whether or not the patients were male or female. In terms of balance, I'm trying to think along what dimensions the data might be imbalanced. I mean, there could be situations where, there's very few patients with small tumors, let's say, and then there's, majority of patients with big tumors, or vice versa, right? I think that for the types of problems that we were looking at here, I don't think data imbalance was a big issue, but certainly I could imagine in other related applications it could be a problem. But I think the kind of standard tricks of dealing with imbalance on the prediction side in terms of sampling or boosting or things like that could help. But it's a good question. I don't know how it would perform after you do the prediction and then you run it through an optimization model, whether or not kind of the attempt to rebalance would be kind of maintained when you run it through the optimization piece. So I think that's a great question, but I don't really know the answer. Okay. There's another question here. We mentioned that our study reached the high 70s. How high is the threshold for implementing this in the real world? Yeah, great question. I mean, so in the real world, the implementation was in a sense 100%, right? Because when we were looking at all of the plans that were implemented, the metric that I was showing you is how close did we come to meeting what was actually done. So we basically reached 80% out of 100. What I didn't show you is, if you think about a 2x2 confusion matrix, right, I'm only showing you one part of that matrix. There's, other criteria that we would have met that the clinical plan did not meet. And so on the whole, I would say our plans were probably on par with what the clinical plan performance was. However, I will say that there's many, clinical considerations that we may not be considering in this model. Like we're looking at kind of the core criteria, but it still relies on the clinicians to look very carefully at like the spatial distribution and perhaps other metrics that they might tacitly review that aren't explicitly mentioned in our criteria. So long-winded response to your question is, I think, in our range, probably 80 to 90%, if we were thinking about it that way, is probably sufficient for implementation, but it really does depend on having the clinical perspective and agreeing that these plans are sufficiently good. Okay. Sorry, I know we're starting to run behind a bit. Okay, so very quickly, this question, since it's a follow-up, what index can be determined to definitively declare that this technique is better than the other techniques? I think that's I don't know that there is such a definitive metric that one can use. The real metric, I suppose, would be long-term outcomes. Can we measure the plans by this model versus plans by that model on 5-year survival or 10-year survival outcomes? So that's the true measure of goodness, I suppose. But for the type of numerical work we do here, I mean, I think that's pretty impossible to have as an endpoint. So really the types of metrics that we are looking at, to what degree do we satisfy the criteria? To what degree can we meet the criteria and lower the dose? It is a bit of a multi-criteria evaluation. We would use that type of criteria to decide what models are better. Okay, thanks for these great questions. So I'll keep going. So as I mentioned, the motivation for this was, everyone is using private data. How can we rally the research community around a common data set and having everyone benchmark their models against using common metrics? So this first competition focused on this part of the pipeline, this part of the KVP pipeline, which is just the prediction phase. So this is going to look very similar to those, international machine learning competitions or Kaggle type competitions that you hear about, right? So the objective was to really spur development and comparison of accurate dose prediction methods on a large open access data set. And then all of the models were trained and tested on the same data and evaluated with the same metrics. Okay. So this might not look large for those of you who participate in these competitions and so on, but this is already would be at the time, I'm pretty sure the largest data set in this space, and it still might be. I haven't checked recently. So we took our raw private data set. So this is the same 217 from before. We took a raw public data set of images. So these images exist on the Cancer Imaging Archive, so anyone can get this data, but these images don't have the dose distribution associated with it. Okay, so what we did was we ingested this raw data. We did a lot of cleaning, and there were lots of things that unfortunately, lots of images that we had to exclude because they were not consistent in terms of like the organs that they included or excluded. The labeling was inconsistent. So we tried our best to clean and standardize as much as possible, but there were some things that we just couldn't fix. So we had to get rid of some incomplete data. We then took these images and we applied the model that I just talked about in the previous part of the talk to generate a synthetic treatment plan for these new images. And so that's the plan augmentation step. So we generate the synthetic data now. And we didn't, again, try and tune or do anything. We just kind of ran it through, and we judged many of them to be low quality. That means that they would require additional tuning to make it better. And we decided we didn't want to spend the time there. We run a tight timeline. So we threw those out and we just kept what was remaining. And then we split that back up into the private data that we started with and the public data now that has now been augmented with these artificial plans. And so the private data got stored away. We never look at it again. And then only this cleaned and augmented public data did we use going forward. And this data set we split into 200 for training, 40 for validation, and 100 for testing. Okay. So how did this proceed? We made our training data available on this online platform, Codalab. We would give everyone access to this data, and we would let everyone develop whatever machine learning model they wanted for dose prediction. And then during the validation phase, teams could make submissions to our competition website, and there would be a leaderboard. And these submissions would be evaluated on the validation data. So everyone sees the training data. Everyone has the X and the Y. For the validation data, obviously, they only have the X. Right? And so they would apply their model to the X, they would generate a Y, they would submit that, we would compare it with the true Y, and then we would calculate some sort of error metric, and then we can rank basically everyone's submission. And this was on a public leaderboard. Okay. Every team, that was the validation phase. You could submit as many as you want. You could see where you are on this leaderboard. And then we would enter the testing phase, where we would say every team could make their one and best submission, and then their one and best submission was applied to the testing data now. Okay, and again, so another data set that no one has ever seen. And then we would get the score, and then we would compare the scores, and we would determine the winner. Okay, so that was the process. We decided to actually have two leaderboards. So there was a dose score and a DVH score. So this would simply be like some percentiles in the dose distribution. And the reason was that the dose score would be a more general metric. So this would be a variation of an MAE, a mean absolute error, on a voxel by voxel level. So a pixel by pixel of each pixel in the image, essentially. Okay. Whereas the DVH score, this is a metric that is much more classical and is very specific to radiation therapy. And we thought that there could be some people who are using more classical methods that would want to participate. So we gave this score as an option as well. And this would be another type of an MAE error measure, but based on the dose volume metrics. So some points on the histogram, we would compare those points and how close those points on the histogram are, and then compute an error that way. Okay. And so we would allow two types of submissions. We would allow people to submit doses, like full 3D doses. And if you submitted a full 3D dose distribution, we could calculate this score. But of course, we could also calculate the histogram. We could generate the histogram and calculate the DVH score. If you only submitted DVH submissions, we could obviously compute this one, but we couldn't compute the dose score because we can't recreate the 3D dose distribution. Okay. Yeah, and then there's some way we calculate it. The score is not that important for the purpose of this talk. But if you're interested, I'm happy to answer questions about it later. Okay. So here was the timeline for the competition. We would make available the training set, and we would start the validation immediately, the validation phase immediately on February 1st, 2020. In April 2020, April 25th, 2020 is when the testing phase would start. We would give people, what is that, like 10 days to submit their testing model. And then we would close the competition. We would notify the winners, and then we would present the results at the major international conference for medical physics of the American Association for Physicists in Medicine. Okay, so that was the original plan. If you notice the dates, you know that something happened that disrupted those plans, right? So COVID happened right around this time. And so all of these dates got pushed, but surprisingly, when we look in retrospect, the dates did not get pushed by that much. And, you know, everyone adapted pretty well, and people kind of continued on with the competition after obviously an initial period of massive disruption. So dates got pushed back about a month, but not really more than that. So the testing, the competition, the winners notified, all got moved only about a month. And because we couldn't make the conference anymore, we presented the results in a webinar that fall. Okay, but I mean, really a testament to the resiliency of the individuals who are involved in this and really wanted to keep participating. So when we started, there were 195 people who were registered. 73 of those people made submissions in the validation phase, and then 54 in the testing phase. And again, much of this drop-off could have been due to COVID, or could have been due to people not being able to get good models running. Some people were a part of teams. So of these 195 people, there were actually 129 teams. So some teams had multiple people. 44 teams submitted to the validation phase, and 28 teams, unique teams, submitted to the testing phase. Okay. And so just of the registered participants, just to give you an idea, we got representation from 28 countries. Over 60% had actually not done KVP research before. So we did some surveying. 57% said medical physics wasn't, their area. So we got a lot of people who are from engineering, from computer science, and like machine learning researchers who had discovered this competition and simply wanted to participate. So we thought this was a nice byproduct of it, to actually try and bring in new people into this field and get them interested in this area of research. Some demographic information, just I think it's kind of interesting, but we did want to look a little bit about diversity of the population. And what we notice is, compared to the standard the general US population, the science and engineering subpopulation, and then looking at our open KVP population, we were much more skewed in terms of gender, in terms of Asian race, much more so than the general population, but no major skew in terms of disability. Okay, so how did the competition unfold? So these plots basically plot the improvement in the score, kind of the mean score, and then the best score over time as the competition proceeded. And this is normalized basically by the total number of submissions that we got over the course of the competition. So we do see, pretty rapid improvement in the mean score over time, and then starting to slow down. But the best scores were kind of pretty steadily going down, fairly slowly. Same type of trend for the DVH score. Okay, but generally, people were improving their models over time, seeing how they would perform and making tweaks to get better. This is the leaderboard for the 2 dose and DVH leaderboards. The team that won first place actually won first place in both scores, which was quite impressive. The team that was second in the dose leaderboard actually turned out to be not so good at the DVH one. And the 4th place team in the dose leaderboard actually was second best in the DVH score. And so we basically awarded 3 winners of the competition, which happened to be Team 1, Team 2, and Team 4, basically the first, and then the 2 other teams that finished second in each category. So quickly to summarize this, so this was the first competition and first open data set for KBP. When we did some analysis of the team's submissions, every single team that entered was using a neural network-based model, perhaps not so surprising in retrospect. We saw that lots of data augmentation methods around flipping and rotations and cropping images, and a lot of ensemble type methods were very common amongst the submissions. And we managed to work out with the kind of a flagship journal in the field, medical physics, to publish like a little mini special issue around the competition. So we published a paper that described the competition and all these results that I just showed you now. And then the top 3 teams were also invited to submit a paper describing their method as well. And so that, subsequently people continue to build on the models that they developed. And now all of the competition code and the data is freely available to the public. It's hosted on this GitHub. And we encourage you, if you're interested, to look into it and potentially to continue pushing this research area forward. Okay, and then so in the last few minutes, I'll just talk briefly about the Open KBP 2.0 competition. So this one wasn't a competition like the first one was, but we were really thinking about extending beyond the prediction now to the optimization part of the pipeline, okay? And so instead of having a massive open call, we said, let's do it a bit more targeted. Let's invite the teams that submitted predictions to the testing phase of the Open KBP 1 competition. And because this is a bit more of a specialized part of the competition, we said, let's just look at kind of the best, the people who were predicting doses at the very end of the KBP 1. Let's ask them if they want to participate. 21 of those teams said yes. And then we took those predictions and we would run it through a set of optimization models to do benchmarking as well. So 21 teams said yes. 2 of those teams' models had dose predictions that were not as good. And so in the results that we show and the results that we include, we don't show those results, but we still include all 21 teams in the study. Okay. We developed 4 optimization models. So the details aren't so important, but we developed 4 optimization models. And what we are going to do is run each one of these prediction models through each one of these optimization models to get a huge number of treatment plans so that we can try and understand, what's the relationship between prediction and optimization and the quality of the plan.
There is one more question.
Sure. So the question is, was there any internal validation of the predictive model's performance done after retrieving the public data? That is a great question. I don't think, let me think. I can't remember now. I'd have to go look back at the paper. On the surface, I don't know if we explicitly looked at this, but I think we did take a kind of a snapshot just to quickly look at some of the best predictions that were coming in and to see how close they were to the ground truth. And they were basically, if I recall, I think the performance was on par with what we were expecting. I mean, I don't think we were getting like, we weren't, on a scale of 1 to 100, if we were at 80 before, I don't think we were getting hundreds. I think we were getting in the ranges of the 80s in that range, because I think there is a fundamental limit of how well we can do this prediction. So yeah, I'd have to look back at the paper to verify though. Thank you. Okay, so for this part of the so this OpenKBP 2 competition, we did the same thing we had before, right? We had the training and validation data. We trained these prediction models. So we have these dose prediction models from, these would be the ones that the 21 teams submitted. And of course, we only used 19 of those models, if you recall. So then we would get these predicted clinical doses, and then we would run them through 4 different optimization models, and they would each produce 4 different plans. So 1,900 models, we had 100, if you recall, we had a 100-person testing set. So that was 1,900 dose predictions. And each one of those predictions went in 4 models. So we had 7,800 plans, corresponding to 78 distinct pipelines. So 19 prediction models times 4 optimization models. Okay? And we then evaluate these plans using the same criteria metrics as before, criteria satisfaction metrics. Okay, and so here is a quick snapshot of the results, and there's much more analysis in the paper, which I'll point you to. But here we rank the 19 models in terms of decreasing performance in the dose score. So this would have been the best, the model with the best dose score, the second best, and so on, of the ones that submitted. Okay, and these dots, so here I'll just pick an example. So the blue is the prediction. When we take the prediction and we run it through one of these optimization models, we typically get performance that improves, because the optimization is finding a way to improve essentially upon what the prediction thinks is the best possible dose, because now we're doing actual optimization, so we can actually have slightly better performance. So for example, this prediction was at around 70%, and we got it up to around 75%. Yeah, so here, I guess to the question from before, I guess everyone was more or less in the mid 70s, which is, pretty close to where we were before. I think we were in the mid to high 70s. So this is pretty on par. So nothing significantly better than we had before. Yeah, and so yeah, one of the takeaways is that optimization can improve predictions, and even in some cases, right, when we look at like the 16th worst prediction model in terms of the dose score, it still was able to, the prediction was pretty low, but we could actually still generate pretty good plans after we've run it through optimization. And this has become a theme in some of this research, where when you bring prediction into an optimization model afterwards, even a bad prediction could still result in a good solution post-optimization. Okay. But basically, we looked at this and we said that, there's all of, for many of these cases, the prediction models were quite compatible with the optimization models that we generated. Okay. I noticed there is the GitHub link has a virus. Yikes. Okay, thanks for pointing that out. I did not realize that. This is, I'll have to, let me write that down. I'll have to email Aaron. So this is on Aaron's GitHub, but I will make a note and let him know. Thank you for that. Sorry, on slide 33, the 10th team is better than the first one. Yeah, so here, oh, you mean for the blue dot? Yeah, so the blue dot is right. So this is ranked on dose score. Dose score is a measure of essentially MAE. Okay, so this is basically descending in terms of MAE, you can think of. But MAE is not this blue dot. This blue dot says what fraction of the satisfied criteria is this plan satisfying. So just because you have a lower MAE does not necessarily mean you will satisfy the same number of criteria sorry, if you have a lower MAE, doesn't mean you'll actually satisfy more criteria than another one, because it depends on where you are relative to these thresholds, right? You can have many that are just off by a threshold by a little bit, or you can have many that are very far off from the threshold but satisfy a bit more. So, but in general, there is a trend that, you know, higher MAE will give you a lower dose rank. Sorry, a higher MAE, which corresponds to this higher rank, will give you a lower satisfied criteria, but it's not perfectly monotonic. Okay.
And there's another. Oh, I guess
that I think that answers that question as well. Yeah.
Yes.
Yeah, so just a quick summary for this one. So this was an international effort to evaluate the full pipeline, the prediction and the optimization. And so we showed that these pipelines often had better clinical criteria satisfaction than the reference plan. Sorry, I forgot to mention that this dashed line was the reference. So the kind of average across all of them was about 70%. Okay. And again, the code and the data to reproduce these results are available on GitHub. I will make sure that Aaron checks this GitHub link to make sure it's virus-free. And the point is that, again, for this research was to try and really catalyze future research on the optimization side. Up until now, most of it was really focused on the prediction, but the optimization and we're trying to advocate for is really an essential piece of the knowledge-based planning pipeline. And you really need to look at how the prediction and optimization work together in this research. Okay, so just one slide to conclude. I thought this was a really rewarding journey. It started with an excellent master's student who was Aaron, and then he became a PhD student. And then this really took off when we paired Aaron with another PhD student who had, and the two of them really had complementary expertise in medical physics, optimization, and deep learning. And that's where all of the progress that was made on these projects that I talked about today were had. We believe very much so in reproducibility and open science, and part of the goal of having these competitions was to try and putting the data out there and putting the code out there was really to try and encourage this type of behavior. And finally, you know, we think it's also important to bring new ideas and new scholars into the field. And so when you have these open competitions, right, and people say they join this field from a brand new field, we like to think that it actually brings in some new ideas into the field and helps push things forward. Okay, so thank you, everyone. That's my presentation for today.
Thanks a lot, Tim. This was great. I see no other questions. So on behalf of everyone, I'd like to thank you for your time. I also like to thank our colleagues from the University of Seoul for co-hosting this seminar. And I encourage everyone to check our website for the upcoming seminars in the next 7 weeks. Thanks,
so
everyone.
Yes. I don't want to cut it short, but we're happy to end it here. So thank you everyone for coming. If you do have any follow-up questions, I'm happy to stick around for a few minutes and answer questions, or of course you can email me. I'm happy to talk offline as well.
Yeah, feel free to type in your questions. Oh, there is a question.
Sure. So there's a question around the optimization method. Could I explain it a little bit? Yeah, I tried not to make this talk very mathy. So I wish I had my iPad right now. So maybe I'll talk about this optimization method. So the optimization model is, so this is this would have been, I think, a linear programming model, this would have been a linear programming model. So it would be a model that looks something like, I don't know, say this. So we would have a matrix C where the rows of C correspond to different objective functions, so different cost vectors. So that C transpose X, you get a vector, and then you would do alpha transpose that vector. And so you would think about this as weight. So this would be like alpha 1. Think about it as like alpha 1 F1 of X plus alpha 2 F2 of X, where these F's are linear functions, essentially. Okay. And so this is what we're minimizing, subject to a bunch of constraints around dose, around the deliverability of the fluence, that basically the intensity of the radiation beam and so on. So if you knew what the right values of alpha were, like, and this basically would say things like, this would be the importance of this first objective, alpha 2 would be the importance of the second objective, and so on. And so these objectives would be things related to the dose criteria. So we want to deliver a lot of dose to the tumor. We want to deliver not very much dose to the brainstem. We want to deliver not very much dose to these spinal cord, and so on. Okay. And so this process here, the inverse optimization process, is to learn what these alphas are. So we have an initial model that's based on duality, where we learn these alphas. And once we learn the alphas, we plug it into this model and we solve this forward optimization of minimizing, alpha transpose this function subject to a bunch of constraints. So that's at a high level what the optimization model is doing. And if you want more details, maybe we can talk offline, or I'm happy to just point you to the papers and you can see all the details in the paper.
And two final questions I see as well.
So the first question is, what would I propose as some future research in this field? So I think that's a great question. I mean, there is a lot of, there continues to be a lot of interest in, let me clear this. And so this has been studied a lot. This has also been studied a lot historically. I think the kind of interplay between these two has been studied less so. So thinking about how we link models together in a way that kind of generates the best solution. So I think that's an interesting avenue of research. I'll also say that there is another approach that has already been developing for quite a while now, which is image into a neural network into a treatment, right? So bypass this two-step process, directly do an end-to-end model where you can take an image and you generate a basically these these beams and these intensity profile across the beam from different angles and so on, right? I haven't looked at the latest literature on this in a while. I think this research has had some successes, but I think there's still more that you can do in this area to make these predictions directly. I'm also, or these inferences directly. I still feel like the kind of, a method that is more similar to the traditional approach. This would be a bridge between the traditional approach and like a direct neural network inference. Like, I think this intermediate approach, I think, still has value because, people talk about interpretability and things like this. Like, I think there is some amount of interactiveness a clinician can have with this type of a pipeline that is still important, I think, to get buy-in and to get to a solution, rather than having a neural network generate a solution from scratch. And then if it's not good enough, it's kind of unclear how you change it to get a better solution that you like. So anyway, all that to say is I think there's much more work that can be done in thinking about how to make this pipeline more efficient, how to make it better, how to make it more tunable, how to make it acceptable to humans. I've only talked about one treatment modality here, which is intensity modulated radiation therapy, so delivering static beams, right? There's a version of this where we're delivering radiation kind of 360 degrees, as this thing is rotating. So that requires different models. We could extend it to different cancer sites. I think there's a lot in that direction that can still be done. A question from an anonymous attendee, Professor Chan, do you accept PhD students for working on research in this area this year? The short answer is yes, I will consider PhD students applying to work with me on this topic.
Are there any more questions? Feel free to type them in. In the meanwhile, I'm gonna put in the chat box the link to our events for you to check our upcoming events in the following weeks. And I'd like to thank everyone again for attending tonight, and for Professor Chan for the great presentation, and thank our colleagues from the University of Seoul for co-hosting this seminar series.
Okay, thanks everyone. Thanks for taking the time out of your night to be here, or out of your morning. And yeah, I hope you enjoyed the presentation.